I want to talk to you today about some of the work that my group does with bats, and I'm really glad to be here. Thank you, Alex, for the invitation. And I want to tell you in particular about some of the work that we're the most excited about right now. So this is going to be a mix between finished work that's published and that you could read if you were interested, and also some of the ideas that we're chasing that we don't have full results on yet, but that I'm just enjoying playing with. But before I start, I would like to acknowledge that I'm speaking to you today from the traditional and unceded territory of the Algonquin Nation in Ottawa, and that my team also works across the traditional and treaty territories of a number of First Nations. And the reason that that's important for our particular work is that we try to understand and conserve threatened wildlife. And the reason that we here now can do this now is because of millennia of effective care for the land and our wildlife relations across Turtle Island by Indigenous people that allowed those species to persist. And in particular, I would like to acknowledge that although we're trying to do our part in my lab, which is mostly a lab of settler scientists, to contribute to wildlife conservation, the real contributions to wildlife conservation in what is now Canada are coming from Indigenous-led stewardship. And I think that's what is going to get us where we need to go. I think I don't need to go into detail here because Alex has already given you the rundown, but I was until recently a government scientist with the government of Ontario and built a lab up at Trent University. And then in 2021, we picked up and moved over to Carleton, which is where we are now. And I'm going to share a few of the stories of our work, but I wanted to just shout out to the amazing students and postdocs and collaborators that I get to work with, because I am incredibly lucky and grateful that I have such a wonderful team to do science and just life with. So let's start by just a quick outline of the kind of research we do, and then we'll dive into bats in particular. The goal of our research is to understand the impacts of rapid environmental changes, such as habitat fragmentation and wildlife disease and invasive species, on small or declining wildlife populations. And wildlife health is a key focus of this work, which falls into these four interacting themes that I'll walk you through. So we do some of our work on host pathogen interactions and disease ecology. And in particular, we've been doing work with bats and white nose syndrome, which we'll talk about today. Also some work with chytrid fungus and amphibian populations, and with ophidiomycosis or snake fungal disease. And then also some work with coronaviruses, specifically bat-borne coronaviruses. And we work with diverse taxa, so reptiles, amphibians, bats, sometimes plants, sometimes fungi, sometimes birds. I'm a reluctant bird biologist, but we basically select the system that's going to be the most effective for answering the questions that we have. Whoops. We also work to understand connectivity and conservation of aerial habitats, this field of aerial conservation, which is adjacent to aerial ecology. And we want to understand the impacts of habitat modification and fragmentation on endangered species. And all of that feeds into this idea of developing and evaluating effective tools for species recovery and habitat restoration. So our work is very applied. Although we answer fundamental questions, we always have a goal to inform a new policy or a new policy direction that could be recommended. So what do I mean when I talk about One Health? There isn't a single accepted definition of One Health. Instead, it describes a perspective that recognizes the interconnectedness of people and animals and plants and their shared environment. And so studies of pathogen and disease are part of a One Health approach, but we apply a broader definition to wildlife health here that also considers other determinants of health, so access to resources and the impacts of other stressors beyond disease. And so this is also very interdisciplinary. We work with health scientists and with veterinarians as well, so that we can pull on a broader toolbox than maybe we have ourselves in my conservation ecology lab. And my students and postdocs and I do most of our work in this really heavily settled landscape of Southern Ontario. So I'm on this landscape right now, and so are you. You have some idea what I'm talking about. Many of the sites that we work in are just these tiny remnants of suitable habitat for these tiny remnant populations of endangered species that are endangered largely as a result of landscape modification over time, right? Like this Blanding's turtle that's living in a wetland that's shrinking in size and it's polluted with pesticide runoff, but it's still there. And so we're working to keep the remaining populations as viable as possible and to ensure persistence and hopefully growth and recovery over time. And the thing that we can get from a One Health approach is to acknowledge that this turtle's health is also tied to our own. And so what both we and the turtle need are science-based solutions and policies that recognize that connection. And going back to my land acknowledgement and Alex's land acknowledgement, the idea that everything is connected is in no way coming from Western science. Western science has just taken a really long time to get there in an effective way. So I recognize that this is not a new idea, but this application of multiple determinants of wildlife health is relatively new to Western science, and it's an idea that we're playing with a lot at the moment. So we can also apply One Health approaches to bats, right? We can think about how this endangered tricolored bat is partly affected by disease and partly affected by other stressors. And I want to talk about some of those threats in detail, but first I'll do a really quick introduction to bats for those of you who aren't familiar yet. So what do you need to know about bats to follow the rest of this talk? First, they're totally addictive. Secondly, they're the only flying mammals. They're in their own order, the Chiroptera, with over 1,400 species described so far. You can see that this big brown bat is wearing a band on her wing that allows us to recognize her when we recapture her. And so we can apply mark recapture analyses to track population trends. And that's a large part of what we do. Another thing you need to know about bats from a conservation ecology perspective is that bats are not flying mice. In fact, they have very little in common with mice or other rodents, most rodents. So bats can be quite long-lived. The bat that Einstein's holding here can be over 40 years old. So older than Einstein, until recently older than me. And that's a real contrast to most small mammals, and especially rodent small mammals that tend to have very short lives with very high fecundity, but bats, along with their long lifespan, have very low fecundity. So this female little brown bat gave birth to a pup that came out at about a quarter of her body mass. So that's like me giving birth to a 40-pound baby. Don't picture that. And they provide really intensive care to these giant pups. You can see this bat is nursing her gigantic baby still. Soon it will start to fly on its own. And then if she is in good condition the next year, she might have another gigantic pup, or she might skip a year if she needs a year to recover. So really long lifespan and very slow recruitment to populations. And we'll revisit those traits again because they're critical for bat conservation and for recovery planning. So let's take a look now at some of those threats that the bats in Ontario and in Canada face. I presented some of this work in a seminar in 2018 here, and so I'm not going to repeat the details of that work, but we'll build on some of it. As you know, bats in Ontario are threatened by white nose syndrome, at least the species that hibernate are. They're also threatened by a range of other diseases, and many of those are diseases that we haven't even identified in bat populations. Sorry, I just got the McAfee pop-up window, which is really great timing. Oh dear, technology. How far can I wing it? Give me 1 second. Okay. Cross your fingers. Okay, wonderful. We're on. You've had time to ponder white nose syndrome and other diseases, which we'll talk more about. Some bats are also affected by collisions with wind turbines, which create additive mortality for already threatened populations. Roosts can get disrupted, especially if they're in a woodlot that's getting cut down, or if they're in a building where the human co-occupants of the building don't really want to have bats there. We have general habitat loss and alteration that can affect bats. Pollution is probably a threat, but it's one we know very little about. Climate change is impacting all of the species on Earth, and bats are not immune to this. We think that some bats may be faced with declining prey. We hear, big statements about declining insect abundance globally and in particular regions. And although I know there's controversy about the details of those analyses, there is good evidence that in some regions there are declines in insect populations. If you're an insectivore, that's a problem, right? And then of course, there's all of the threats that we haven't even realized are there yet that we're not able to study effectively. And each of these needs to be considered on its own to first understand it really well, but we also need to consider the cumulative impacts of these threats, and those can be additive or interactive. And the short answer to what are those cumulative impacts at this point for bats in Canada is that we barely have enough information to understand each of these threats in isolation. But we are trying to start to tie them together in a more One Health informed approach to understand overall how our bat populations are doing. So if we start from the disease side of health and we look particularly at white nose syndrome, you already know that this is a disease of hibernating bats, right? And we have 5 hibernating bat species in Ontario. And white nose syndrome is caused by a fungal pathogen called Pseudomonas destructans that was introduced to North America in or just before 2005, 2006. It's native to Eurasia, and it causes this fungal growth on the bats during hibernation that can also cause disease. So it was introduced, if you can see that red X on top of the blue shaded squares on this map of parts of North America, in New York State, in Schoharie County, and then you can see that it spread over time from that point of introduction. So the shading here is just showing you spread through the subsequent years from 2006 all the way to the present. And the most recent records from Canada are those ones right on the border of Alberta and Saskatchewan, which were actually just publicized last week. So you can see it's been spreading over time, and it has some pretty major impacts on some of the bats that it encounters. So I should pop that arrow up before. So generally what happens is the fungus grows on the nose, that's where it gets its name, also on the wings and on the ears, and damage to the wings disrupts homeostasis during hibernation. And you get this cascade of physiological effects that can include dehydration, starvation, and eventually mortality. But it's really important to understand that not all bat species are equally affected by white nose syndrome. And to understand this better, I designed a series of studies looking at gene expression in response to infection to understand how different bat species respond to the fungus, and also how the fungus responds to different bat species, because I had this hypothesis that maybe the fungus does something different on tolerant and susceptible species. Spoiler, it doesn't. But the take-home message here is that tolerant species of bat in Europe that co-evolved with the fungus mount a very similar immune response to the very susceptible little brown bats that we have here. Even though they're closely related and mounting the same immune response, we saw high mortality in the North American species that was previously naive to the fungus, and no mortality in the European species that had co-evolved with the fungus. And the similarity of that molecular response. So first we thought maybe they're doing something different, but they're not. The similarity of that response gave us hope that there was potential for evolutionary rescue in the little brown bats here, if they could come through a similar bottleneck to the one that the European bats may have also experienced. And we needed that hope because things got pretty bleak. So the initial mortality from white nose syndrome in little brown bats was over 95% at some sites. This is a picture just showing bones on the floor of a hibernation site where many, many bats had died from white nose syndrome. And that was really grim. But the question I was interested in was this one. If we have 95% mortality in a population of little brown bats, but 5% survive, what's the difference in the survivors? And first, I approached this question from a genomics point of view. Like I said, we looked at the molecular response of different species to try and understand if the survivors were doing something different, if susceptible and tolerant species were doing something different, controlling for phylogeny. But we also wanted to understand what happens after this big selective sweep from white nose syndrome, right? Because it's a huge selective pressure if you wipe out 95% of the population. And so one hypothesis was that white nose syndrome was exerting selection on immune genes or on other heritable information, that it could tell us which kinds of recovery actions we could prioritize. So imagine that you have a population of bats and they have different immune gene variation among individuals. So let's say each color of bats on this banner represents a different immunogenetic profile, and it's produced by previous selective pressures on the population that have created the variation we see today, right? So let's say we now bring in a novel pathogen like white nose syndrome that causes 95% mortality. We have two different scenarios that we could find, right? Scenario one is that the disease is not exerting immunogenic selection. Scenario two is that the disease does exert immunogenic selection. If selection is occurring, or sorry, start on the other side. If selection is not occurring, if we follow scenario one, then we'd conclude that the adaptation to the pathogen is unlikely, right? We would see the same frequency of the different variants in the surviving persisting 5% that we saw in the initial population before that selective sweep. And we conclude adaptation is unlikely because nothing seems to be predisposing these bats to surviving, right? These seem to be maybe lucky stragglers that just haven't died yet, or something else is involved with survival other than the immune genes we're targeting. If we look at scenario 2, where disease is exerting immunogenetic selection, we would predict that in our sample of bats that have survived the disease, we see a higher frequency of some particular type of genetic variation that predisposes them to surviving, right? And this would tell us that adaptation to the pathogen is possible, that we want to aim for supporting evolutionary rescue. And the reason that this matters beyond academic interest in disease ecology is that in the first scenario, we would want to focus on treating the disease, right? We'd want to slow that extra mortality as much as possible and hope that somehow we would find a tool to mitigate the disease and we could prevent extinction. But in the second scenario, we'd want to focus on supporting recruitment, right? We wouldn't want to start treating the disease and slowing this process of selection. We would want to support reproduction by those survivors because they are carrying a heritable trait that they can pass on to their offspring that will help them survive white nose syndrome. So we tested which of these two scenarios play in a study where we designed a sequence capture assay and we targeted 138 immune genes and their regulatory regions, which we hypothesized might be involved in white nose syndrome survival. And we compared immunogenetic profiles of little brown bats that were sampled from Manitoba, east of Prince Edward Island. If I mirror it on your screen, I've done that backwards, but you get the picture. And they were sampled before and after the arrival of white nose syndrome. So we could look at variation geographically, but we could also look at variation in immune genes in bats that were sampled before white nose syndrome and bats that were sampled after. And we found signatures of directional selection by the pathogen on little brown bats. And this is the first study with direct evidence of immunogenetic selection by white nose syndrome on bats, and suggests that there is something different about the survivors. And that's great news because that tells us that evolutionary rescue may be possible. And recent work by other groups following up on this have also supported this hypothesis that evolutionary rescue is ongoing and that there may be potential for recovery of these populations over time. But remember that they're long-lived and they're very slow to reproduce. And we'll come back to that again. Of course, white nose syndrome might also exert selection on other traits too. So for example, skin chemistry may be involved. So Karen Vanderwolf just finished her PhD with us. She's now a postdoc at UWaterloo down the road from you. I can't talk. Waterloo down the road from you. And part of Karen's thesis was characterizing the skin chemistry of bats that were more or less susceptible to white nose syndrome. So she did the first characterization of pH variation across the wings of bats. And she's interested in understanding whether skin pH is also under selection by white nose. And all of this work looking at specific traits and suggesting potential evolutionary rescue is also supported by our field data, which is, again, good news. So if that 5% that survived were just bats that hadn't died yet, but they weren't less susceptible, we'd expect to see an ongoing decline in our population counts, right? But I'm thrilled to say that's not what we see. So Lauren Hooton, who's also working on her PhD with us, led a study using winter bat count data from hibernation sites in local caves and abandoned mines. And here are some of the results. So on the y-axis here, you're looking at the number of little brown bats counted in the hibernation sites. And on the x-axis is the year of the census. So we put on our Tyvek suits and we get all geared up and we climb underground. Some of you have done this work as well. And the red line here is showing you the year that white nose syndrome arrived at these sites. And you can see that there's that characteristic 95% decline. Everything's terrible. It's tragic. It's, you know, eco-anxiety runs high. The thing that I want to draw your attention to, though, is that we have recent counts of hibernating bats that are actually tracking upwards. And we missed a few years of counting between moving among jobs and rules about whether we were allowed to go underground or not. But in our recent counts, we do see this slow, steady increase. And we'll do another one this month, and hopefully we'll see the same trend. So we do see this general trend of recovery, but now we're into classic conservation ecology, small population land, right? And you know that very small populations are much more vulnerable to demographic and environmental stochasticity, and that threats that might not be that severe for a large population can suddenly become a problem for a very small population. So what are some of the other threats that these populations are facing? Well, for one thing, white nose syndrome is not the only disease that the bats have to deal with, and the fungus is not the only pathogen that they encounter. This is a study that was led by Sonu Subudi, who did his PhD with Vikram Misra's group. And Sonu characterized this persistently infecting coronavirus in hibernating little brown bats. The thing that is good news about this particular coronavirus is that it didn't cause clinical signs of disease that we know of in bats, and no clinical signs have been described as of yet. The question we had about this coronavirus is that it's now overlapping with the fungus that causes white nose syndrome in some bat populations. And so the question was, what if the bats with this coronavirus also get white nose syndrome? Will there be an effect on their immune response? Either their antiviral immune response that helps them suppress viral activity so that they don't express disease, or on their response to white nose syndrome. And so we did this experimental study where we had a control group of bats that did not carry the virus and were not infected with the fungus that causes white nose syndrome. That's the pink group here. We had a co-infected group, the green group at the bottom, that had the fungus and the virus. And then we had a fungus-only group shown in blue here and a virus-only group shown in yellow. And we found that co-infected bats expressed more severe clinical signs of white nose syndrome. So if you were a bat carrying the virus, you were happily bombing along with no ill effects from the virus, but then you were infected with the fungus, your white nose syndrome was more severe than it would have been otherwise. We also found that bats that were co-infected had a 60-fold increase in viral shedding compared to bats that only had the virus. So the response to the fungus, the immune response the bats mount against the fungus to fight off white nose syndrome, requires them to downregulate some of the antiviral responses that they're so good at that would typically suppress viral activity. And this has all kinds of interesting implications for potential spillover, not just of coronavirus, but if this applies to other viruses as well, then potentially for things like rabies virus, which our bats also carry. So if we put all of these disease studies together and we think about what it tells us for bat conservation, because that is our ultimate goal, the recovery actions we'd recommend for bat populations that became endangered because of white nose syndrome should consider impacts of co-infection and not just the impacts of white nose syndrome. And they should focus on maximizing survivorship in affected populations because it looks like the survivors have heritable traits that predispose them to survival that they can pass on to their offspring, and also focus on maximizing recruitment to these populations for the same reason. And based on what we know so far, we think that this is a more effective approach than focusing directly on white nose syndrome, which is a threat we're not really good at mitigating. And in fact, trying to mitigate the threat as white nose syndrome continues to spread might reduce the strength of that selective suite, which may be what's required for evolutionary rescue in the long term. So moving away from disease for a second, if you are a little brown bat and you've survived white nose syndrome and you can tolerate the disease and you emerge in the spring and you're ready to produce your gigantic baby, what do you need? You need safe roosts and you need foraging habitat that's safe and filled with lots of insects, right? And there's a direct link between protecting bats and protecting wild habitats that support healthy, diverse insect communities. Wetlands, for example, which are currently under fire, but that's a different talk we can do another time. Anyways, going back to bats. The problem is the bat's roost site is currently defined as habitat under provincial and federal policy, and it can receive some protection under the existing policies. But the foraging habitat, the airspace where the bat takes over and spends most of its night feeding, and the air that the bat flies through to get from its roost site to its hibernation site and back again, that habitat is not recognized or protected in policy. And of course, many threats to aerial species are not on the ground or in the water. Often wildlife is killed while flying, right? By collisions with wind turbines, by cars, collisions with buildings, collisions with airplanes. But again, aerial habitats aren't explicitly recognized in existing policy. And this isn't a problem specific to Canada. This is a problem worldwide. So even the Berne Convention on Migratory Species doesn't explicitly recognize the airspace as habitat. So I teamed up with some colleagues to think about possible solutions, and we started with trying to define aerial habitat because that is the first step to having it recognized in policy. So we proposed a series of habitat definitions for the airspace that were analogous to those used in marine systems, where habitat is also defined by depth. And this cartoon illustrates some of the threats that are faced by aerial biodiversity in the airspace. So we've got a very general selection here of wildlife that uses the air, and then a general representation of the heights that different threats might face. And we argue that some of this is, some of these effects are habitat fragmentation, that some of these factors are actually fragmenting aerial habitat. And if you move higher into the airspace, which planes do, so if you go above basal aerial habitat and way up, you'll still find all sorts of wildlife. There's bats and songbirds that go 2 to 4 kilometers above the ground. Some geese fly ridiculously high. If you go high enough, you find bacteria that live in the lower stratosphere that have evolved to live there and have increased UV resistance and all sorts of other really neat adaptations. So does it matter if we name these habitats or not? And it turns out that it does, because we protect migratory pathways for many aquatic and terrestrial species. But for aerial species, their habitat's not defined until they land. If you look up, for example, on the IUCN website, it's the International Union for the Conservation of Nature, defined habitat for Carleton's warbler, which is a migratory species. You'll see defined habitat in Michigan, and then defined habitat down in the Caribbean where it overwinters. There is no connected defined migratory habitat. So still, I often run into people who think that this is not a problem and they're skeptical, and that's fair. So here's a different illustration of what migratory aerial species are facing, assuming that it will play. So in red here, you have airports, and in blue, we have cities. And this is what a migratory bat would be flying through, flew from, say, New Orleans up to Ontario. White, as we get further up, are wind farms. Black is roads. Purple is FCC communication towers. And the point here is that if you were a bat or a bird or a dragonfly and you were trying to navigate this gauntlet without going above 1 kilometer in altitude, you would have a hard time doing that in a straight line, right? So you would need to adjust your migration accordingly. I can see the chat lighting up. I'm not watching it yet. Alex, can I just get a thumbs up that you can still hear me okay, and I should keep going? Things are good? Okay, thank you so much. Okay, so if we think specifically about the kinds of habitat fragmentation that bats here might be facing and that might be a threat to them, we know that wind turbines fragment aerial habitat. And we know that the number of wind turbines in Ontario, for example, has been increasing steadily. And that's a good thing. This is not a story about wind turbines being bad in any way. We need renewable energy sources to get us away from fossil fuels and to meet our needs as a society. And that wind energy is amazing, but we need to make decisions to build renewable energy capacity that doesn't threaten biodiversity. And we can do that thing. So in Ontario, bat mortality is monitored at wind turbines for the first 3 years of each project, and then the results are shared with the provincial governments if the work is done on provincial land. If it's on federal land, then the results go to Environment and Climate Change Canada. So the work on provincial land has produced this really large data set, which I was able to access and analyze in a collaboration with the Government of Ontario, Environment and Climate Change Canada, and a really excellent consultant in BC named Kelly Squires, who I can't say enough good things. If you ever get a chance to work with her, please do. She's awesome. So we collected data from this big database from 48 facilities and 594 turbines. And our big question was whether the abundance of bats in the airspace was declining over time. Some of the species that we were looking at are affected by white nose syndrome. Some are more affected by collisions with wind turbines. And we figured, well, maybe this wind turbine mortality data is a good way to get at questions about changes in abundance over time. And our logic was that we monitor fisheries by throwing nets out in a controlled way and quantifying survey effort of netting. And in fact, wind turbines spinning through the airspace at known times and for a known number of years are doing the same thing. They're sampling a piece of the airspace, and so they are unintentionally fishing for bats, right? So we used the numbers of mortalities documented at wind turbines to look at changes in abundance in the airspace over time. And here's what we found. We found that the captures of bats by turbines decreased over the 7 years of the study. All of these decreases were statistically significant, and the steepest decrease was in silverhead bats, which decreased by 91%. The smallest decrease, and I'm putting smallest in big quotes here, was in big brown bats, which decreased by only 65%. So the important thing to note here is that none of these 4 species have been assessed yet by COSEWIC, which is the Committee on the Status of Endangered Wildlife in Canada, although 3 of them are up for assessment in April. So they're not considered species at risk. And these data suggest that these populations, which are supposedly fine, may actually be in decline. And although we didn't have sufficient data for the models for endangered little brown bats, because the models wouldn't converge with what was available, we did see a qualitative decline in apparent abundance of that species as well. And part of that is probably from the effects of white nose syndrome, but it's possible that wind turbine mortality is also having an effect on little brown bats alongside the effects they experience from white nose syndrome. We did a quick follow-up study on this led by Ali Anderson, who was a postdoc with us. And Ali found that the height of the turbine and the amount of area swept by the blades are both associated with the levels of bat mortality. And that's important because turbines are getting bigger and bigger and bigger over time as the design of the turbines is improved. And bigger turbines are more efficient. And again, we need renewable energy. Bigger turbines on their own are not a problem. But when we don't use effective mitigation for bat mortality, the bigger turbines also increase the likely effects on bat populations. And the good news is we know how to fix this. This is something we can fix. What if we did use effective tools to slow or stop mortality at wind farms? If we turn off the turbines and stop the blades from spinning from dusk to dawn, even just during peak migration period, we see a 90 to 95% decrease in bat mortality. So we can have bat-friendly wind energy. But let's say we did that for the wind turbines. What else are the bats facing? If we go back to this picture, and I'm not going to replay the animation, but if you just think about this for a minute, you can see in blue the cities, large cities that are mapped on here, and you can see that migrating bats or birds are going to run into quite a lot of urbanization, right? And part of that is going to involve quite a lot of artificial light at night. So if you are a bat or a nocturnal bird, you're going to encounter these other effects as well. And they may also affect your use of aerial habitat. So we're going to continue working on these questions with bats, but I'm going to jump into birds just for a second. This is the eastern whippoorwill. This is the closest thing you can get to a bat and still be a bird. So we'll just call it an honorary bat with feathers. And the reason that we did this particular study with whippoorwill is because they're big enough to carry the tags we needed to use. So this is Alicia Korpak's PhD research. She's based at the University of Manitoba, co-supervised by Kevin Fraser. And she put GPS tracking devices on whippoorwill in Manitoba, and my team and a collaborator at York University put them on whippoorwill in Ontario. And then we released the whippoorwill and let them tell us where they were going. So this is just a whippoorwill taking off to do whatever it is that whippoorwill do. And after recapturing the birds the following year, we could pull locations off the tags and find out where they went during the migration. So Alicia used the program CircuitScape to create a map of dark sky connectivity to understand the effects of artificial light at night on whippoorwill migration. So these are pathways that migrating birds might use if they avoid artificial light at night, and pathways the birds might use if they were attracted to the light. So the darker areas are areas with less light at night. The red areas are the areas with the most light at night. And then Alicia used the hundreds of GPS points to retrieve from the whippoorwill tags to test how they respond to light pollution. So those points are shown in with the white dots. And she found that aerial habitats are effectively fragmented by light pollution, at least for whippoorwill. Dark sky connectivity was the best predictor of whippoorwill route use, so the selection of the migratory route. And migratory stopovers occurred almost exclusively in dark rural areas. And the signal was consistent when she repeated the analysis at multiple spatial scales. So we're following up on this work now with bat tracking. And again, this is part of Lauren Hooton's PhD work. We've also just brought on 2 postdocs to work on this next piece. And I don't have results to share for this next piece, but I did just want to share the ideas because, again, we're really excited about them. So you can see the antenna coming off the back of this bat. It's got a little tic-tac size transmitter that Lauren's glued right between the shoulder blades. And she's looking at connectivity within a network of swarming sites that we've been monitoring as part of our long-term marker capture work. The piece that we're really excited about at the moment is tracking bats, not just around swarming sites like these ones, which then also become hibernation sites. You can see that in the picture on the right. But also around maternity sites. So we've got a set of maternity sites within the catchment of the swarming sites. And our goal here is to achieve full annual cycle tracking of bats, understanding how the bats from a particular maternity site move to swarming sites and back again, how they move from the hibernation sites to the maternity sites in the spring, and also the important movement corridors among those various roosting sites, because this can help us characterize critical aerial habitats as well and understand how bats are using these. And this also lets us test all sorts of other questions that I can just geek out about forever, like, is there socially facilitated migration among these sites? The bats are going from a point source maternity site to a particular hole in the ground for swarming, which they may then also hibernate in. Do the juveniles from this maternity site follow adults to get to that site? How do they know where they're going? This work also lets us keep integrating disease ecology in our work. So an ongoing project that I'm super excited about is tracking the movement of native bat coronaviruses through these populations and understanding what factors drive transmission of coronaviruses through these networks that we're characterizing. And so this is collaboration with Samir Mubareka and John Kotwa at Sunnybrook Health Sciences. And we are surveying bats for a whole suite of different coronaviruses. We've already found several, and hopefully we'll be able to publish those results soon. And then the next step is to link the disease prevalence data together with the movement data and the social networks that we're able to build based on our tracking and marker capture work. So maybe next time I can tell you about that. I'm really excited about it. It's good that I can't tell you more about this right now, because I want to use the last few minutes here to tell you about one more threat that's looming on the bat's horizon. So we talked about all these different threats and how they interact, but we haven't really talked about what happens in the bat's foraging habitat if we don't fragment it with cities and wind turbines and so on. Is that enough? Is that all we need to do? Is the bat's foraging habitat secure otherwise? There are all of these big splashy studies out there about global insect declines, or at least regional insect declines. And I'm aware that there's argument about how accurate some of those estimates are, and those are legit. But there does seem to be increasing evidence that, at least in some regions, insect abundance is declining. And so that would have an impact on the bats that we work with, because they're all insectivorous, right? And so to try and understand some of these questions, we did a study with collaborators in Yukon. So the study area is shown here, and it's got Nahanni and Nasicho on one side and Kloani on the other. And Brian Slow and Pia Kuka and Tom Jung were our collaborators up there, and they've been monitoring a network of bat maternity sites and very generously offered to share their data with us. And then Val von Zuben assisted with the analyses. She's based in Peterborough, and Brian Gerber at Rhode Island helped us with some marker capture work. And so what we did was we took the bat data that Tom and Pia and Brian had collected, and luckily for our purposes, bat biologists always measure forearm of the bat and then they take its mass. Forearm measurements are messy. They're not always super accurate, but they can still be informative. And mass, again, depending on the accuracy of the scale, can give you an idea of the bat's mass at the time of capture. And because we had this data over a good time series, we were able to look at changes in body size over time. The length of the forearm tells us about nutritional access as the bat was developing, so nutritional access as a juvenile, and the mass of the bat at capture tells us about its recent access to food. So this was a long-term marker capture set, 10 maternity colonies, 14 years of data, and over 3,500 unique individuals. So we removed recaptures and we used a set of linear mixed effects models to test the predictors of bat body size. And in this case, we looked at effective day of year to look at seasonal variation. We looked at reproductive status, and we also tested some climatic variables. And the big picture take home here is that we found average mass and forearm length both declined over the study period. The decline in average forearm length suggests constant addition of slightly smaller bats to the adult pool. And again, this is telling us about nutritional access while the bats are developing, so in the juvenile stage. This set of graphs here is showing you the results for mass. And again, mass is telling you about more recent access to nutrition. We have adult females on the left and juveniles in purple on the right. That decline in adult female mass looks very shallow, but it was significant over time. And the decline in the juveniles, we only see for the last part of the season. So if we just zoom in on that graph for a second, you can see that the two darker purple lines, which represent earlier captures in the season, are actually increasing or stable. These are captures when the juveniles are still being supplemented by their mothers. And so if there is nutritional stress affecting the mothers, they seem to be able to offset the effect on the juveniles through lactation. But that light purple line is captures of juveniles near the end of the season, once the bats are foraging independently, and the mothers are no longer able to supplement. And so they're experiencing the same effects that the mothers are. And again, we see that decline over the 14 years of the study in average mass. And this had links to weather as well. So mass declined with increased rainfall, and rainfall is increasing. This is the proportion of precipitation days. So if a bat experienced a particularly rainy season, it was on average, had a lower mass than bats caught during a drier time. We think this is because it's hard to forage by echolocation in a rainstorm, because the echolocation signals bounce off the raindrops as well as off the prey. So bats typically suspend foraging when it's very rainy, and their foraging opportunities decline. It's possible there's also an effect of decreased insect abundance in this data set, but there isn't a robust data set on insect abundance for this region that we could use to test that directly. But we hypothesized that that might account for some of the variation that wasn't explained by rainfall. Overall, the bats are getting smaller. Does that matter? Who knows? So we did a mercury capture analysis and estimated survival probabilities, and we found that the only predictor in our data set of survival probability was forearm length, and bigger bats have a higher survival probability than smaller bats. So yes, it does seem to matter. The reason this links into our One Health approach is because this is a population in Yukon where the individuals are getting smaller that has not yet been exposed to white nose syndrome, and white nose syndrome selects for bigger individuals. So these two selective pressures operating in different directions might leave those bats kind of crunched in a situation where they're not able to adapt the same way that the bats in Eastern Canada have adapted. And so this is a good example of how we can have a disease pressure that can be exacerbated by other stressors that are also acting on a population. So do we need specific policy tools designed for bats to try and recover endangered bat populations based on some of these results? And of course, there's great related work coming out of lots of other groups, including, of course, Quinn Weber's group in your department and some of the bat labs down the road from you. If we think about what all those studies tell us, do we need specific bat-related policy? Well, identifying habitat more effectively will help. So some of that full annual cycle tracking will help. I mean, I could geek out forever about the social effects of, social network effects on disease transmission and all these other things, but those aren't necessarily getting us to a clear policy for bat recovery. In general, a policy approach that's likely to support bat recovery is pretty anticlimactic. Are you ready for it? If we think again about recovery actions for endangered bat populations, we should consider cumulative effects, and those could be additive or interactive. We should focus on maximizing survivorship and recruitment. We've talked about this already. The good news is this all lines up really well with our own needs. So if we come back to where we started from, we need the same things that the bats need. They need healthy insect communities, and so do we, because those provide really important ecosystem services to us. They need healthy habitat for roosting. We need healthy wildlife populations that are less likely to spill over wildlife-borne pathogens to humans, right? And we also need healthy natural habitat that we can use for the things that people need. We see potential impacts of climate change on bat health, but of course, any action we take to reduce climate change to benefit the bats will also benefit us and every other species that we share the planet with. So I'm going to stop there and just thank my funders and thank you again for the invitation to join today.